Hey guys, Brown here, back at it again. Going to learn a little bit more about neurons. In the previous video, we kind of just barely glanced over the surface about what makes up a neuron, so I thought I would kind of get a little bit more into the weeds about the three main parts of a neuronal cell, that is, the soma or perikaryon, the dendrites, and the axon. So we're going to start with the cell body or soma or perikaryon or whatever you want to say. And this is a transmission electron micrograph of developing brain tissue in a mouse. So you can kind of see some, this is a capillary over here on the right hand side. So these are blood cells here. We're mostly interested in the developing nervous tissue over here on the left. So we're actually going to focus in on this top left-hand corner. So if we enlarge that, enhance, we can see some of the features of the perikaryon or soma of this nerve cell. The first is, of course, obviously the nucleus. And the thing to notice here is the relative lack of heterochromatin. Neuronal nuclei are very transcriptionally active, and so we see mostly new euchromatin, not new chromatin, Patrick. Oh, lordy, euchromatin in the nucleus of this cell. We also see a Golgi apparatus. So that's right over here. There's a Golgi. The Golgi tend to be perinuclear and somewhat large compared to what you're going to see in other cells. Tons of mitochondria. Neurons are very, very, very energy-hungry cells. When you sleep at night, you're still burning tons of calories because your brain is still firing. So these cells are very energy hungry, and so you're going to have lots of mitochondria down in there. And then finally, and it's probably a little bit difficult to see on the video screen, but over in here we have some developing neurofibrils. And neurofibrils are special cytoskeletal networks within nerve cells that are going to help with with transport. So some of these nerve cells can be really long, 100 centimeters or more in length from soma to the tip of the axon. And so we have to get stuff from the trophic center of the cell here around the nucleus out to the tips of those axons. And these neurofibrils are going to help. In the light microscope, they're argyrophilic, meaning they stain readily with silver stains, and they're a combination of both intermediate filaments and microtubules. Now, there are a couple of things that we can't see very well in this transmission EM, so let's look at a couple of light-based micrographs. This image on the right is actually a stacked confocal image superimposed on top of a DIC micrograph, but there's structures down here at the tips of these white arrows, this kind of pinkish stuff in here. These are called Nissl bodies. And Nissl bodies are actually stacks of rough ER, kind of lamellar rough ER, so it's layered like pancakes, plus the polyribosomes on top of the rough ER. So if we were staining in, say, the light microscope, or transmitted light microscope, we weren't doing confocal, and we used a basophilic dye like, say, Crescel Violet or something like that, these would be very, very, very dark blue staining because of all of they're just very basophilic because of all of that nascent protein being made. On the left here, at the end of the arrow, we see some aggregates of a material known as lipofusin. Lipofusin, when viewed in unstained tissue, has kind of a brownish color to it, and it's actually the remains of old dead lysosomes. Okay, so the cell body, it's where we find not only the nucleus. Notice all the euchromatin in this nucleus. Even in a light microscope, you don't see much heterochromatin. So we've got a nucleus, we've got the Golgi, we have tons of ER in the form of Nissl bodies. We have the lipofusin, which is the used up lysosomes. I can't make words today. And tons and tons and tons of mitochondria. So now let's go out and look at the dendrites and look at some of the specializations that allow dendrites to do their jobs. So this is a micrograph we've seen before. It's a pyramidal cell. And we see numerous dendrites out here on the periphery of the cell. And one of the things to notice about dendrites is that typically there are many per cell. Like we said before, multipolar neurons are the most common neurons. But dendrites tend to branch. If we look at this one here, we can see as we move away from the cell, it's branched. And then this one branches here and here. And then this one comes up here and branches again. This one is going to branch this way and this way. This branch comes like this. You get the idea. It's mixed, lots of branching. The term for this branching is arborization. You guys know what we celebrate on Arbor Day, right? It's all about trees. And this branching pattern is very similar to how tree branches look. So arborization means to branch like a tree. And one of the things you'll notice is that as arborization proceeds, the dendrite gets thinner and thinner and thinner. So the more branching that happens, the more narrow the diameter of the dendrite as we get further away. You probably can also tell, even from this light micrograph, that this dendrite is covered in numerous spines. So if I look at this picture here, this is a computer-generated simulation of a particular dendrite, so of a single dendrite and all of its spines with the synapses mapped on it. So dendritic spinal synapses are indicated by red, and then straight-on dendritic synapses are indicated in blue. And we can see that these dendritic spines are going to increase the surface area of the dendrite vastly, which means we can have more synapses. You can have up to 10,000 dendritic spines on a single cell. That's a lot. And as I said before, these dendritic spines are going to give us more surface area for more synapses, which is going to allow this neuron to communicate with more upstream cells. Like more cells are going to be able to talk to this neuron because it has all of these dendritic spines. And then finally, let's look at the axon. So whereas most cells have many dendrites, you're usually only going to have a single axon. So here it is running away from the cell, very fine. And unlike dendrites, which get thinner and thinner and thinner, axons, although there is some tapering that occurs, tend to be largely uniform in diameter throughout the length of the axon. Axons can be really long. I know you guys have only ever seen face cam of me, but I'm actually 6 feet tall, and that means that I have axons who have a soma in the base of my whose soma is in the base of my spinal cord, and the axons extend all the way out to the muscles in the tips of my toes. You know, so we're talking a couple hundred centimeters here. So that's a really, really long process from a single cell, which is going to require some specializations that you'll see in the module following this video. The axon begins at a structure called the axon hillock. So this kind of pyramidal area here, that is the axon hillock. Now, if we look at this transmission EM on the left, we can see the transition from soma, which the people who made this image have colored orange, to the axon hillock, and axon itself is colored in green. So this is called the initial segment. It's not really super important, but the initial segment is just the axon hillock and the proximal portion of the axon. And you'll notice that in the axon hillock, we don't see any Nissl bodies. So here is a TEM of a Nissl body. Here's some Nissl bodies up in here, but we don't see any of those here in the axon hillock, so that's a good way to spot it in addition to its shape. Now the axon, because it is so functionally, biologically unique compared to the rest of the cell, like muscle cells, it's going to have some funny names. So we don't call the axon cell membrane, it's not the plasma membrane anymore, it's the axonal membrane or the axolemma. Likewise, the cytoplasm inside the axon is very different from the cytoplasm up in the soma, so we refer to this as axoplasm. It's just kind of like we saw in muscle cells. You're going to use a special name to indicate that this has a unique biology compared to other similar things. Finally, the axon is not a single structure that travels from axon hillock to synapse with no deviation. In fact, the axon can and often does branch many, many, many times. So this cartoon that you can see on the left here is a reconstruction of a single pyramidal cell, much like the image that you see on the right, and all of the dendritic and pericardial components have been colored red, and all of the axonic components have been colored blue. And you can see that this axon is going to branch many times, and these branches that come out from the axon that are going to travel to other cells are called axon collaterals. Uh-oh. For some reason my pen, there we go. I think I was hitting a button. Anyway, so these axon collaterals are going to come off to the sides and allow this axon to communicate with numerous other cells instead of just like maybe one at the very tip of the axon. So we saw soma as the trophic center of the cell with the bulk of the organelles. We saw dendrites with their dendritic spines so they can receive input from many upstream cells. And we've seen the axon, which is going to conduct information away from the soma toward other cells. And it's going to use axon collaterals to allow this single cell to communicate with multiple downstream cells. So that's it for now. Hope you had fun. Study hard. See you later.